possible without a computer because you have two different experiments. They can only talk to each other, the crystallography and the electron microscope, through the computer, and the computer makes it possible. Now, this whole achievement involves a simulation constantly of the ribosome that is matched to the data, and that requires computer simulation, a huge computer simulation. If you add the water to the system, you have 3 million atoms. And so you need computer power. And this power is given to us through the GPUs in the following way. We are using the supercomputers at the National Science Foundation centers, in particular also the one in our own town, NCSA, and they have a computer cluster called Lincoln. And each of the nodes in this computer has 2 GPUs. And those GPUs can take over some of the computational load, and that you see on the right-hand side how they do this. And they could increase the speed of the simulation that I show you here by a factor of about 3, which is not so great yet, but molecular dynamics simulation is a difficult calculation, very inhomogeneous. It's almost the opposite of what is really good for the GPU. But we are striving and improving it, and actually in case of other proteins, not quite as big, we are getting a speed up of about a factor 10. And we want to work with NVIDIA in the next generation in the next release of our program, namely to improve the situation further. I've been promised by Jim Phillips, head programmer of NAMD, that this capability of NAMD using GPUs will be available in the next release that is going to come out actually next month. So this is definitely a great improvement. You just add a few GPUs, and very cost effectively you can increase the speed of an existing cluster then by quite a good factor. My next example is actually also very exciting. It is about genetics. Genetics is not only important for us when we ask the question, how do we inherit our own properties to our offspring? That works, of course, through the genes. But genetics is also important during our lifetime in our body, because the body cells are duplicating themselves all the time. And some die and new ones come, and that has to work. And actually sometimes it doesn't, and then it's connected with bad diseases, particularly with cancer. And scientists have realized that at this stage of genetics, it is not just the genes that are playing a role, but also properties that do not come about through mutating the genes in our body. That can happen, and that leads then also to diseases, but it can happen very systematically. The body has a mechanism by which, for example, our diet, our level of exercise, other life experiences are altering our genes, not through mutation, but rather through methylation and other chemical changes. And here you see the change to methylation. You add on the right side, you see the famous Watson-Crick helix in blue, the normal one, and then in case of what is today called epigenetics, life experiences lead to the methylation of DNA, but it's a very, very subtle change. The change just involves at certain few spots on the DNA to replace a hydrogen atom by CH3, a carbon surrounded by 3 hydrogens. It's just a very small pimple on the DNA every once in a while. And in fact, when you look at methylated DNA that is biologically relevant, you realize that its physical properties are almost not changed. Very, very difficult actually to measure methylation profiles, because there's such a subtle change. Now, we work together with Greg Timp now at Duke University, earlier at Notre Dame University, earlier at our university, and together we realized the following: that if we bring the DNA into a situation where it is mechanically stretched, so that it becomes stretched out and that its strand partially separate, then the DNA that is methylated shows actually real differences. And you see that on the right side, you see the simulation of a DNA going through a nanopore that is in the actual experiment even narrower than they are seen, so that the DNA can hardly go through. And if you have normal unmethylated DNA, you need to squeeze electrically the DNA through the pore, and you need a voltage difference of 3.5 volt. Whereas if you methylate the DNA, it's easier to get it through. It only requires 2.5 volt. So this is very interesting, because for the first time we see a real difference. But what is absolutely exciting is the explanation that is given by simulating the same process correctly in the computer. You see there the two types of DNA, and you see first that the DNA that goes through the pore is actually stretched out. It is not twisted helically as normal DNA, but it is forms more like a straight ladder, and the bars of the ladder are very ordered and neat in case of methylated DNA, whereas some of the bars are missing and even twisted outside in case of non-methylated normal DNA. And so here we see for the first time a difference, and that difference is very obviously important because strands, DNA needs to be untwisted, strands need to be separated when you actually want to read genetic information in the cell in order to express it in terms of proteins. So this is a wonderful result from a simulation. But what I want to point out in order to come to the role of the GPUs is something more mundane, but also of great practical importance. You see there on the top the nanopore, and this is a very, very tiny pore. In this case it is a 1 nanometer wide pore, and they can easily get clogged. It can actually be clogged by the ions that are transported through the pore along with the DNA. And that was observed, that you're getting this pore clogged all the time, and of course the nano engineers want to prevent it, but they don't know what's happening. Simulation shows what is happening. Here you see one of those precipitates, a clogged pore, particle in the clogged pore, that is doing the clogging. And it is actually the result of a simulation. The simulation shows exactly how this plug forms and also how it can be dissolved. The problem is only that it is very difficult to identify, to see the plug in a simulation, because the plug has almost the same density as the liquid surrounding it. The only difference is that the atoms in the plug are more like a solid. They don't move relative to each other, whereas the ones in the liquid are liquid and they move much more disorderly. Now it turns out that physicists know how to see the difference through what is called the radial distribution function that looks at the relative order of atoms. And you see on the right side that a precipitate had a very different radial distribution function than a liquid. And so one can identify the difference. The reason is only that to calculate radial distribution functions is very, very costly, because you have to look at the relationship of all the atoms in the system, and that requires a lot of computational work. There come the GPUs. The GPUs can do it much, much faster. We found an algorithm that could do it. This kind faster, beta is to the top. And you see this is a logarithmic scale. You see that we could turn a 15-hour computing job into a 10-minute computing job. And we could also make good use of the latest GPU chips, NVIDIA chips. And so we have basically solved the problem now for us to detect clogging or precipitate formation is really important. You may frown a little by saying, oh, this is really little primitives to look at clogging of some pores. But actually it's very important because these kind of issues play an essential role in nanoengineering, and the computer is an ideal microscope for nano devices. It can visualize them where other methods cannot see them. Many methodologies, for example, work only in dry samples and not in fluid samples, or they don't work with organic samples. And that is where computing can help. Now, the cell is also doing lots of chemistry. And chemistry involves to rearrange the electrons in a molecule. And this turns out to be a heaven for GPUs. Electrons follow the laws of quantum physics. And the laws of quantum physics and GPU computing go hand in hand wonderfully. They love each other. And so in fact, we heard a lecture yesterday from Harvard University, and here's an example from Stanford University. Quantum chemists learned to use GPUs to do their job much faster. But here we have now a new problem. Today we can compute not only the motion of molecules, but also along with the motion of molecules, the motion of the electrons. But to view those motions is very, very difficult, because basically you have to calculate the clouds in the sky. There are many, many shapes, and to do this mathematically is very, very difficult. But we achieved to do it with the GPUs. You see here a buckyball vibrating on the left, and on the right you see the electrons moving along. Visualized in real time, visualized as quickly as you see now the movie in front of you. So a job that took a day, and where you had to do, a few frames, and had to go home and come back the next day, you can now see interactively. And so this will revolutionize chemistry and biology, because now you see finally also the motion of the electrons along with the motion of the atoms, and thereby you see the full reactivity and get the full picture. Why is this so? Why do GPUs help? The nice thing is that you can calculate the electron clouds point by point independently. So you can generate many threads and put them on the GPUs. We also learned to put the data that are needed to calculate those clouds that are given by quantum chemistry program into the memory of single GPUs by starting with horrendously huge files that we realized contain huge amount of redundant information that we condense down to a minimum of what is actually all the different information needed, and then we started to compute. And lastly, but very, very importantly, the GPUs are very good in calculating transcendental functions. And those are the functions like exponential, sines, and cosines that are needed to describe the quantum mechanics of electrons. And so this all together, as you see here on this table, gives us a speed effect of almost 400. And so this is really ringing the bell for a new era, the era of molecular electronic dynamics. Now, my last example is protein folding. That is an example that is connected with accuracy of simulations. Here you see a protein called Willen headpiece. We stretched it out. In the background we show the crystallographic structure of the protein. And now the computer tries to see how the protein spontaneously folds and refolds. Looks like it's not going. It's supposed to go into the structure. Yes, it went now. So we see here a wonderful example, a great triumph of modern computational biology, that we can fold proteins. And that's very, very important for understanding quite a few central nervous system diseases that are connected with the misfolding of proteins. So it looks all great, except there are some beasts, some proteins that just don't fold right. Here is one of them. And so this is about the same size protein, and as we will see, it just doesn't fold, it doesn't fold. And you may think that, Schulten, you are just too impatient, wait a little bit. But we could show that actually it is not due to waiting, or not enough waiting, but rather that it is due to the inaccuracy of the forces